This is the second in a series of videos in which I introduce the biology of cancer. This video is about genes and genetics, and I'll start with a diagram illustrating what I call the core idea of cancer. A normal cell at some time acquires a mutation or other kind of gene change and becomes a slightly abnormal cell. Sometime later, one of the progeny of this cell will acquire an additional mutation or other kind of gene change, and this process will continue with, among the progeny of each cell at each stage, one cell acquires an additional mutation or gene change until eventually we achieve a fully blown cancer which may be malignant and may metastasize. I want to emphasize that these gene changes or mutations are many and varied. We can have simple mutations like a single base pair change in the DNA, or we can have very large mutations, I'll still call them mutations, right up to a whole chromosome getting lost or added to the cell. And in between you can have deletion of a large chunk of DNA or extra copies of a large chunk of DNA or inversion of a piece of DNA and so on. Anything that can happen to change the overall DNA content of the cell and its sequence. But in addition to that, there's heritable epigenetic change. So these are things like histone marks and methylation of DNA that inherited from one cell to its progeny. And these can contribute to cancer gene changes as well. Then there are viruses. Some viruses bring into cells genes which add to this process. For example, the human papillomaviruses, among which are some which cause cervical cancer, can bring in genes which change cell cycle regulation. And finally, there are mobile elements, which are stretches of DNA which can replicate themselves and insert new copies of themselves into other parts of the genome. Finally, we have to consider how many of these gene changes are necessary to get a fully malignant cancer. And for a typical human adult cancer, we think the answer now is 10 or more. It's a lot, and it's a lot more than people imagined in years gone by. A very important idea in cancer biology is each mutation gives the cell that acquires it a selective advantage over its neighbours, so that cancers evolve by natural selection, as illustrated in my rather crude drawing here. We start with a cell acquiring a mutation, and if we come back days, months or years later and look at the tissue where this cell was, we'll find that the progeny of that mutated cell occupy more of the space in the tissue than their neighbours. So this cell has acquired a selective advantage and it gives rise to an expanding clone of cells. Among those cells will be one that acquires another mutation, and that mutation will also give that cell a selective advantage. So again, if you come back days, months or years later, you'll find that the progeny of that cell occupies more of the tissue than its fair share. And this process continues. As I've said, it may continue as many as 10 times until you have your malignant cancer. This whole process of acquiring mutations one after another is evolutionary, and it's often called progression by pathologists. It also, we can see, leads to the idea that tumours are essentially clonal. This used to be academically a very interesting and controversial point, but we can now say with confidence that most tumours are essentially clonal, in that virtually all the cells in the tumour will have almost all the same mutations which they've acquired along the way. And every time you do an experiment in the lab where you look at the mutations in a tumour or you look at the proteins it produced, you will find that most of the cells have the same mutations. This process of clonal expansion as the result of mutations that give cells selective advantage can be illustrated experimentally. And I would like to show you a beautiful experiment done by Maria Alcalia, Phil Jones and their colleagues a few years ago. It's illustrated here. We start with a normal control. What they did was they arranged for a few cells scattered around in a mouse esophagus to have a fluorescent label, a yellow fluorescent label as it appears in these pictures. If you follow the progress of these cells over a year, what you find is that some of these individual cells divide and give rise to a small clone of cells of 2, 4, 8, 16 or whatever cells, whereas some die out. So at the end of a year, there are fewer clones, but some of those clones have several cells in them, so that the overall proportion of cells which are fluorescently labelled in the esophagus is the same as it was at the beginning of the year. So on average, these cells haven't acquired a selective advantage. We're merely looking at a stochastic variation in the number of progeny from these cells that survive. They then introduced an additional mutation into these cells, as well as the fluorescent marker. For those of you who know developmental biology, it was a dominant negative mutation in the notch pathway. And you can see that the pattern of development here is completely different. At the beginning, the esophagus has a few scattered fluorescent cells, as in the control above. But as we go through the days, months and years, more and more of the esophageal surface is taken up by the fluorescent cells, until at the end of the year, essentially all the cells on the surface of the esophagus are expressing the fluorescent marker. So these cells acquired a mutation which was introduced by the experimentalists, which gave those cells a selective advantage and gave rise to clonal expansion. A final comment about this clonal expansion process, if we go back to our diagram of the developing tumour and think about what this final tumour will look like, if we take a sample of tumour from a patient, we should remember that it will often contain some normal cells and it will contain some remnants of clones which were present in between. So it will be a mixture of the final clone, the final cancer, and some of its precursors which may be present as well as some possible normal tissue. The next big idea is that cancers, or at least adult cancers, are usually genetically unstable. What this means is illustrated if we go back to our core idea of cancer here. We see the process of accumulation of mutations is speeded up by genetic instability. What this means is that the cells undergoing this process are in fact genetically unstable. They're more likely to acquire mutations, or they have a higher rate of acquisition of mutations than normal cells. This in itself, we think, is due usually to mutations or gene changes. So, for example, maybe one of the first or second mutations in this sequence is a loss of, shall we say, a DNA repair enzyme by mutation of its gene, or something like that. Some kind of mutation or gene change which makes the cell more liable to accumulate mutations. And this accelerates the whole process of acquiring all these mutations. It's been known for a long time that cancers often have quite a lot of damage to their genome. For example, they often have very messed about chromosomes. But it used to be thought this was just kind of something that happened along the way because the cells were dividing very frequently and they sort of tripped over their chromosomes, and that this wasn't really very important. We now know it's a much more specific issue, and the evidence for this is very straightforward, that different cancers have completely different patterns of genetic instability. And I'll show you some early examples of experimental evidence for this. Here we have the chromosomes of a colon cancer. It's a thoroughly nasty, fully malignant colon cancer, but it has completely normal chromosomes. We're using a technique here in which chromosomes are stained a different colour using fluorescent dyes, according to which chromosome they are. So you can see here there's 2 copies of each chromosome, and they're all stained in a different colour. There are a few odd little dots and blobs which don't quite match up, but that's just technical limitation. So this particular tumour has normal chromosomes, but it has an increased rate of small-scale DNA mutations. If you measure the rate at which mutations occur in a particular enzyme, you can show that it's literally 100 times the rate that you would expect in a normal cell. My second example is also a gut cancer, but in this case the chromosomes are all over the place. The number of each chromosomes is variable, and many of the chromosomes are made up of multiple pieces taken from different chromosomes. You can see a chromosome here which is made up of several different pieces of several different chromosomes, shown by the different colours. This particular colon cancer doesn't have a significantly increased rate of small-scale mutation, but it has a defect in the repair of DNA strand breaks, which gives rise to this horrendously reorganised and rearranged chromosome pattern. From a theoretical point of view, there was much discussion of the significance of genetic instability. It was argued by some that in order to get the number of mutations you need for a fully blown cancer, you needed genetic instability. But this was disputed by others who claimed that provided cells were cycling regularly, the normal background mutation rate might be enough to accumulate the number of mutations you needed. I'm not sure whether the jury has really come back on this one, although now that we know that there are a lot of mutations in adult cancers, it does seem more likely that you need genetic instability. But the safest way of presenting this is to say that in practice, the great majority of at least adult human cancers are genetically unstable, and we can see this experimentally. So how does genetic instability come about? There are 2 obvious broad categories of defect that could give rise to genetic instability, illustrated here. You could have a defect in DNA repair, so that the spontaneous damage which inevitably occurs to DNA all the time is not repaired efficiently, and you can also have errors in replication of DNA or in mitosis. Both of these mechanisms can give rise to either small-scale changes in the DNA or large-scale changes. We've already seen examples of failure of DNA repair in the 2 colon tumours I showed. The first one, which has a high level of sequence instability, has a defect in DNA mismatch repair. This not only results in a high rate of single base pair changes, but it also leads to the instability of repeat sequences called microsatellites. And this kind of defect was originally discovered and named microsatellite instability, but now it's more correct to call it a defect in mismatch repair. The second of our tumours had a defect in DNA strand break repair, and in fact it had a mutation in the hereditary breast cancer gene BRCA2. Mutations in the other hereditary breast cancer gene BRCA1 would have given much the same effect. But it's also clear that defects in replication and mitosis are important. For example, it's known that a small proportion of colon cancers, amongst others, have a defect in DNA polymerase epsilon, which is one of the DNA polymerases which is used to replicate DNA. The defect is in the proofreading ability of the enzyme, so that when it occasionally makes an error and adds the wrong nucleotide to a DNA strand, it's unable to check and correct that error. This leads to an extremely high rate of point mutation, something like 1,000 times the normal rate of mutation in a normal cell, and about 10 times as much as a defect in mismatch repair would give you. People have also seen defects in mitosis. And here's a fluorescent picture of a cell in mitosis. It's taken from a video from Dwayne Compton's lab where they followed mitosis and showed that some colon cancer cell lines, for example, had lagging chromosomes. You can see a lagging chromosome here. It's in the middle between the two bunches of chromosomes which are moving out towards the spindle poles. His chromosomes got left behind. It may end up in the wrong cell, or it may end up being pulled apart and broken. This lagging chromosome mechanism has been shown to lead to quite catastrophic changes in individual chromosomes. There are probably other mechanisms as well. So we have examples of all of these four possibilities, and they're clearly more than this. We also know that there can be epigenetic instability. Some colon cancers seem to be unstable in their DNA methylation and acquire a lot more DNA methylation than they're supposed to in the wrong places. And also we have to consider things like mobile element activation, which can give rise to DNA changes. To finish with genetic instability, a couple of final comments. First of all, the actual genetic instability of most human cancers is not yet known. So if you go to the hospital and you find a patient who has a cancer which has just been diagnosed, in general, even if it's genome sequenced, we don't know what the mechanism of genetic instability will be in most of these cases. Another very important point about genetic instability is that it's a target for therapy. Drugs have been developed which target DNA repair mechanisms, for example, so that if a cancer has a defect in a particular kind of DNA repair, it may be extra vulnerable to inhibiting a different kind of DNA repair, which will result in there being so much DNA repair inability that the cell becomes non-viable. And this is shown to work as a therapeutic strategy in cancers, particularly in the use of so-called PARP inhibitors. Another very interesting possibility is that older kinds of cancer therapy, the cytotoxic therapies, may actually inadvertently be exploiting genetic instability. If you think of drugs like the vinca alkaloids, which interfere with mitosis in the mitotic spindle, and drugs like cisplatin, which cross-link DNA, it may be that these have a selective toxicity for cancer cells in some cases because those particular cancer cells are defective in being able to repair that kind of damage or manage the interference with mitosis, for example, that the vinca alkaloids would give. This is a very interesting idea, but as far as I know, it's not yet been fully worked out. It's convenient at this point to introduce the terminology oncogenes and tumour suppressor genes as a way of classifying the mutations that cause cancer. There's more than one definition of these terms, but the ones that most people use and works best is that oncogene mutations are overactivity mutations, making a gene overactive, whereas tumour suppressor gene mutations are inactivation or loss of function mutations. Let's have a look at the consequences of these definitions. An oncogene mutation is an overactivity mutation, and if it affects one copy of the gene, then that gene will be overactive. The fact that there's a normal copy is irrelevant, and the cell will become abnormal. So you only need to mutate one copy of a gene to get an oncogene mutation. In contrast, for a classical tumour suppressor gene mutation, if you mutate one copy of a gene so it's inactive, you still have the remaining normal copy, and in many cases this will be quite adequate and the cell will remain normal. It's only when the second copy of the gene is lost that you get the full effect of the mutation. You get a proper tumour suppressor gene mutation. So tumour suppressor gene loss of function mutations require, in general, both copies of the gene to be damaged. Now, biology is always as complicated as it possibly can be, and of course there's an intermediate case where mutation of one copy to a loss of function mutation is actually quite important in the cell. The other copy of the gene will usually be lost later anyway, because this gives a further selective advantage. There's a very good example of a mutation which is of this kind, and that's in the p53 gene, which is probably the most frequently mutated gene in all of human cancer. The p53 gene encodes a tetrameric protein. And as this diagram shows, if you think about a tetramer, if one subunit of the tetramer is inactive, that may disable the whole tetramer, or at least make it work less well. Certainly if you disable 2 copies of the protein, then the tetramer probably is non-functional. So you can see that somewhere between 1/4 and 1/16 of the protein will remain functional, even if only 1 copy of the gene is mutated. So we believe that's why mutation of p53 in 1 copy does seem to convey a selective advantage, but usually you lose the remaining copy as well. Now, you might think that if you have to mutate both copies of a gene, that's much less likely to happen than if you only have to mutate 1 copy of a gene. But of course it's more complicated than that, because to get a mutation that makes a gene overactive is much more difficult than to get a mutation which simply disables it. I mean, you can delete the whole gene or break any part of the gene and you'll disable it, whereas to get a gene to function more efficiently, you probably have to alter something in a rather subtle way, and we'll get some examples of that in the next video. To illustrate oncogenes and tumour suppressor genes, I'll use the control of the cell cycle, which is of course important in its own right in the context of cancer biology. The cell cycle is conventionally represented as a circle with a series of phases. At the top we have G1 phase, which is the resting phase where the cell's not doing anything very much. And indeed, if the cell remains in G1 for a very long, if not permanent period of time, we call it G0. At some point, the cell decides that it's going to replicate its DNA, which is the beginning of the action. As the DNA is replicated, we go into S phase, S for synthesis. And when synthesis is complete, there's a recovery phase where the cell sorts out any remaining problems and prepares itself for mitosis. This is called G2. And then mitosis itself, the pulling apart of the duplicated chromosomes, is M phase. And then the cell is back in G0 again. At the junctions between these phases, there's often a checkpoint period where the cell kind of looks around and sees whether it's ready to go on to the next bit. And the most important of these checkpoints for us is the G1S checkpoint, where the cell makes the decision to replicate its DNA, which is in effect its decision to divide. The G1S checkpoint is therefore very closely controlled, and a key pathway which controls this culminates in the RB protein. The RB protein holds the cell cycle closed, shut, but it's inactivated by phosphorylation, and that phosphorylation is done by a complex which includes typically a cyclin-dependent kinase, a CDK, and a cyclin, such as cyclin D1. These in turn tend to be regulated by proteins which stop them from phosphorylating RB, and among the proteins which can do this are P21 and P16, shown here in red. So you have alternating stop-go signals as you go down this pathway, with the proteins at the top which stop the kinases from stopping the retinoblastoma gene having the same effect as the retinoblastoma protein. They stop the cell cycle. So if you want to get a cell to divide too many times, what you do is inactivate either the RB protein or the gene that encodes it, or you inactivate the genes for P21 or P16. Since these are inactivation mutations, they will be tumour suppressor genes, which is why I've put them in red. On the other hand, if you want to get the cell to cycle faster, you can make the kinase complex more active, and so making the genes which encode the cyclins or the cyclin-dependent kinases more active is an oncogene mutation. And a simple example of an oncogene mutation in this case would be for there to be many copies, multiple copies of a gene like CDK4. Multiple copies of the gene means there's more of the protein, which means there's more kinase activity. So that would be an oncogene mutation. One of the things that you can see looking at this pathway as an illustration of oncogenes and tumour suppressor genes is that there's no fundamental difference in the activities of oncogenes and tumour suppressor genes, except whether they're plus or minus, if you like. There was a tendency early in cancer biology to treat them as though they were different kinds of gene, and that's misleading. It's much easier to think of all these genes as being cancer critical genes, or as we now call them, driver genes. They all can be mutated to give cancer. Some have to be mutated to be overactive, some have to be mutated to be underactive, but they have rather similar functions and they run in the same pathways. The next major topic is hereditary predisposition to cancer, sometimes called hereditary cancer, although that's rather frowned upon. If we go back to our core idea of cancer, this is easily represented by saying that the first cell in the sequence is not actually a fully normal cell. It already has one of the mutations that we require to get cancer, and this speeds the whole process up. This predisposing mutation, which is present in the germline, is almost always a tumour suppressor gene mutation. What you have then is that you'll have a tumour suppressor gene mutation in one copy of this gene, but not in the other, so that the patient's cells will in general be normal. It's only in cells where the remaining normal copy of that gene is lost that the full effect of the mutation is manifest. So typically what will happen is that, in contrast to a normal cancer where both copies of the gene have to be mutated during life, in the hereditary cancer case only the second mutation has to occur. The mutations that occur in hereditary cancer, or give rise to hereditary cancer, can be either genetic instability mutations or they can be growth control mutations. So, for example, the mutations that we think of as the hereditary breast cancer mutations, the two famous ones in the genes BRCA1 and BRCA2, which are both DNA repair components. The hereditary mutation is inactivation of one copy of either BRCA1 or BRCA2, and this means that when you lose your remaining copy of the gene in one of your cells, that cell becomes defective in DNA repair of strand breaks. I have here some examples of hereditary predispositions to cancer. The first one is a hereditary predisposition to colon cancer, and this particular syndrome is called adenomatous polyposis coli, or familial adenomatous polyposis. But the first form, adenomatous polyposis coli, gave its name to the gene that turned out to be mutated in this hereditary predisposition. This gene is now called APC, and we know that the APC gene is mutated probably very early in the development of the vast majority of colon cancers, whether they're hereditary or not. Next is Lynch syndrome, which is a predisposition to colon cancer and also endometrial cancer, amongst others. It used to be called hereditary non-polyposis colon cancer until it was realised that it wasn't really just confined to colon cancer. So Lynch syndrome is the correct name for it now. It's a defect in mismatch repair, as we've seen in colon cancer before, the two primary target genes being MLH1 and MSH2. Finally, we have hereditary breast cancer predisposition. The two best known genes involved in this are BRCA1 and BRCA2, named of course because this is the way they were discovered. As for APC, mutations in mismatch repair can either be inherited in the hereditary predisposition syndrome or they can occur during life. But rather disappointingly, in the case of BRCA1 and BRCA2 mutations, most of these are germline mutations, and although some people get mutations in these genes during life, it's not a particularly common thing in breast cancer and related cancers. This was a disappointment because when the genes were discovered by studying the hereditary predisposition syndrome, it was hoped that we would find genes which played a key role in all breast cancers, and that hope wasn't fulfilled. That brings me to a question of why we're so interested in hereditary predisposition, and there are two reasons. Historically and academically, there was enormous interest in the hereditary predispositions because finding the genes that were responsible turned out to be one of the most powerful ways of finding genes which are mutated in cancer in the early development of the subject. So genes like RB, APC, BRCA1, BRCA2 were all discovered by studying hereditary predisposition syndromes. But now there's a much more practical reason for being interested in these hereditary predispositions, because they turn out to be among the commonest genetic diseases that are potentially lethal. Let's look at some numbers. I have here two examples of possibly the commonest genetic diseases that are not cancer. Polycystic kidney disease, or strictly autosomal dominant polycystic kidney disease, is probably the commonest known genetic disease that's potentially lethal, and that comes in at least 1 in 1,000 births. Cystic fibrosis is not far behind, with about 1 in 2,500 births in the UK. Compare these figures with the figures for the common cancer syndromes that we know about. Lynch syndrome, which is a defect in mismatch repair, mostly mutations in MLH1 and MSH2, comes in at about 1 in 400 in the UK population. And the incidence of BRCA1 plus BRCA2 mutations together comes in at about the same, 1 in 400. Of these, mutations in BRCA1 is about 1 in 1,400, and mutations in BRCA2 is about 1 in 500. There's also a suspicion that these hereditary predispositions are actually commoner than we realise. Now that we're beginning to sequence the genomes of cancer patients as they come into the hospital, we're finding mutations in genes like BRCA1 and BRCA2 that hadn't been found due to people looking at cancer families where there were a lot of cases of, shall we say, breast cancer in a particular family. So it may well be that the numbers I've got on the board here are a bit low. So you can see that if you're a medical geneticist, hereditary predisposition to cancer becomes perhaps your most important thing to look at. This completes this video on genes and genetics in cancer. We've seen how the accumulation of mutations in a cell can give rise to cancer, and how these mutations or gene changes give the cells a selective advantage over their neighbours, at least in most cases. A typical adult human cancer will have something like 10 or more such gene changes. We've considered genetic instability, which speeds up the accumulation of these gene changes, and discussed some of the many different mechanisms that can give rise to genetic instability. We've considered hereditary predisposition to cancer, where one of the mutations, or maybe more than one of the mutations that create cancer, are inherited. And we've pointed out that such hereditary predispositions are among the commonest of genetic diseases. The next video in this series will consider the cell biology of cancer. In other words, what these gene changes do to the behaviour of cells. I hope to see you there.